welcome everyone to our second webinar in the webinar series for Working Group 2 of the Root Benefit Cost Action. What are we actually about? Let's start with a little introduction. So the aim of this webinar series is definitely just to stay connected with each other and to establish future collaborations as well. So it's really the idea that we get to know each other and all of the different labs that are involved in this working group of the cost action. So what the aim really is that the different speakers will give a global overview of their lab's unique research, their expertise, and our facilities. This is a bi-monthly meeting, meaning that it occurs every other month and at the end of each month as well. So each session takes approximately 1 hour with 2 speakers each. Each have allocated 30 minutes for 20 minutes presentation and 10 minutes questions. All the talks will also be recorded and will afterwards be made available at our YouTube channel. And as you know, we always use this link right here for our platform, which we're using right now. So this link that you can see here, which you all successfully use since we're all here right now. But we're still debating also for the next session if we might use another platform, but for this we will keep you all updated. Then also, if you want to be a speaker once at our webinars, now for this semester we're already fully booked, let's just say. But for the next semester, there's definitely still spots available. So feel free to let Rafa or me know if you're interested in presenting. So as for the speakers we have today, Estibaliz Larrainzar will be speaking as I'm trying as the first speaker, and then we will from Spain, and then we will also have Janis Stringlis from Greece as well. So it will be very interesting to see what the speakers have to offer for us today. So Rafa, I give you the words.
Okay. First of all, First, I'm going to upload the presentation of Estibaliz. Yeah, here. Okay, should be. And give you the. Okay, so Estibaliz, you now should be able to move your slides and everything. So, but first of all, please allow me to introduce Estibaliz. Estibaliz Larrainzar studied agricultural engineering at Universidad Public of Navarra in Spain and earned a master's degree in biotechnology from Wageningen University and Research. During her master program, she had an internship at the University College of Cork, where she first experienced plant growth promoting bacteria. So in 2005, she started her PhD, focusing on the regulation of symbiotic nitrogen fixation in legumes under drought conditions. And during her PhD, she conducted research at Max Planck Institute for Molecular Plant Physiology in Potsdam. And after completing her PhD, she received a Fulbright Spanish Ministry of Education Fellowship to pursue a postdoctoral stay at UC Davis, University of Davis in Douglas, Douglas Cook Lab. She later extended with the support of Marie Curie International Fellowship. In 2014, she returned to Spain to start her own research line on the role of ethylene in regulating symbiotic signaling and nodal function. She has obtained various competitive contracts from the Spain Ministry of Science and Innovation, including Juan de la Cierva and Ramón y Cajal grants. So and now, Estibaliz, the floor is yours. So whenever you want, you can start.
Okay. Thank you very much for the invitation and for preparing this, for organizing this very useful seminars, I think. So the idea, some of you may know me also for my participation as coordinating the training school here in the Root Benefit. I hear some echo. I don't know if it's me. Okay, maybe now better. Yes.
It's okay now. Okay.
Okay, let's see. So today I'm going to present you the work that we are doing here in the lab. Just a brief, so let's see, I need to pass here, right? Just a brief introduction. If you don't know much about us, we are the Public University of Navarra, which is located in the city of Pamplona, that is in northern Spain. We are very close to the French border, close to the Pyrenees. And you may have heard about our city because of the running of the bulls. That is this special festival we have in July. But other than that, the rest of the year, the city is very quiet, and it's really a nice place to visit with a historical city center. And it's really, I encourage you to come. So our group, there you go, belongs to a larger group in a larger department in which several aspects of plant physiology are studied. I will focus mostly in symbiotic nitrogen fixation, and this is an image presenting some of the participants of the group. The group is led by César Arrese-Igor, professor in plant physiology, and myself, that I obtained recently the position of associate professor. And then the work I'm going to present is basically the PhD work of Germán Gómez and the work of Maribel Rubia, who is now assistant professor. And I will briefly introduce the work that is being carried out now by two postdoctoral fellows, Isa Zahid and Lucas Castellani. So, sorry for passing, I need to do this. I think I don't need to introduce much about the legume-rhizobium symbiosis in this conference, but just as a brief reminder, we are focused on the interaction, on the symbiotic interaction with legume plants and symbiotic rhizobium bacteria that develop a structure in the root that is called a nodule, where these bacteria are hosted, and this is a mutualistic beneficial symbiosis for both the plant and the bacteria, in which the bacteria offers a source of fixed nitrogen to promote plant growth, and the plant offers then a source of carbon to fuel the highly ATP-dependent nitrogenase reaction. So what our group specializes is, the core of our group, as I said, is plant physiology. So we are really strong in the characterization of plant responses to a number of conditions, obviously characterization to the efficiency of symbiotic interactions, and drought responses to water deficit. We have a number of methodologies set up to evaluate photosynthesis, respiration, to do kind of targeted metabolomics, so we can measure the content of carbohydrates, amino acid, urates, nucleic acids, organic acid, a number of primary and some of secondary metabolites as well. We have set up for hydroponics, pure hydroponics, and also aeroponics cultures. And one technique that I would like to highlight is the use of apparent nitrogenase activity to measure nitrogen fixation. So you may be very familiar with acetylene reduction assays. That is the most widely spread technique. But here, in contrast to this other methodology, what we do is to measure the level of hydrogen that is being evolved by intact plants. And if you remember the equation for the nitrogenase, there is 1 mole of hydrogen that is generated per mole of nitrogen reduced. So we use this estimation to calculate nitrogenase activity in intact plants. So it's not detached nodules. You can here see the setup. And so we put the whole plant there, we pass a gas flow, and then we measure the hydrogen that is released from these plants. So it's really a useful technique, and it has been proven to be more accurate than the acetylene reduction assay. So the expertise, or the core theme of our group, was the responses of, or let's say the mechanisms regulating the inhibition of symbiotic nitrogen fixation during drought stress that has been worked or studied in the lab for quite some years already. Basically, you could think that maybe the first idea was that maybe the inhibition of nitrogen fixation that occurs in drought stress could be due to a decline in photosynthesis, but this was shown that not to be the case, and actually what we observe is an accumulation of sucrose in nodules, so it's really not an inhibition dependent on lack of carbon substrate. The hypothesis that our group has been mostly focused on is the downregulation of one of the key enzymes for sucrose degradation in nodules, that is sucrose synthase. And this limits the availability of malate to fuel the nitrogenase activity in nodules. So we have been working on this hypothesis and testing this and other hypotheses in several legume plants. And this, as I said, is, let's say, the classical work that has been carried out in our laboratory, and we still continue to do so. When I obtained my PhD, as Rafa has mentioned, I obtained a couple of fellowships to continue my postdoctoral research, and I went to the University of California in Davis, where let's say I took a leap, and then I moved from my mostly plant physiology-based approach to a more molecular approach, and also shifted to early stages of symbiotic nodule development or symbiotic interactions. And this publication was, let's say, the one summarizing my work there at UC Davis, in which what we did was a very detailed time course of early stages of nodulation, or rather of the symbiotic interaction. And with early, I mean ranging from times from 0 to half an hour, 1 hour, 3, 6, 12, 24, up to 48 hours post-inoculation. So those were really very early time points. And at the time, this work represented one of the largest collection of RNA-seq data that was definitely in legumes, although nowadays it has been, let's say, improved by other subsequent work that were analyzing extended time points like Chisel et al. But at the time it was really novel. And we used 4 genotypes in Medicago truncatula, our wild type A17. We used the 2 NOT factor receptor mutants, NFP and LIKE3. And one of the highlights of the work was the inclusion or the addition of the ethylene insensitive mutant sickle. So why I say that this was a great contribution to the work is because, if you may remember, Medicago truncatula sickle is a hypernodulating and hyperinfective plant, a mutant. And this hyperinfective phenotype is due to the insensibility of this plant to ethylene. So it is unable to control, let's say, infection threat development. So having that in our data set, what it did was amplifying the signal that were in a regular route that was only located in a couple of very specific cells. Here we had signal all over the place. Analysis, well, we did also a morphological analysis. So just to keep you, let's say, in a framework, we had our latest time point was 48 hours. So when infection was going on and we're starting the release of bacteria in the cortex. The outcome of this study was the identification of 2 main responses in this data set, ethylene-related responses that we were able to discriminate the results from the mutant, from the sickle mutant and the other mutants. And of course, what we were really interested in, NOT factor-related responses. So the difference between those that were giving infection versus those that not. And then one of the key, let's say, the most selected group of genes that included some of the key markers like NIN, ERN1, and FY1, INOD11, belong to this group with this classical, well, classical now for me, with this typical pattern of expression in which NFP mutants show basically no induction during time. LIKE3 showed a tiny bit of induction because LIKE3 leads a little bit of infection. And then you had a moderate induction in A17 and a huge amplification of this induction, of this expression in sickle. And interestingly, I mean, the data set is there. We are working on some of these genes. Obviously, there are many more for which we don't have a role yet. So this is a nice source of data. You can check, if you have interest, you can check this data set. It's now integrated in the Medicago Gene Expression Atlas, hosted by the people in Toulouse, in which, of course, you have a repertoire of all other RNA-seq data. And if you are interested in a specific gene family of genes, you can send me an email with your list of interest, and I have everything sorted out in R, and I can check whether those are, we can find some members that are differentially expressed, and we can generate this type of graphs. Okay, so what was one of the, or I mean, there were many, obviously many outcomes of this study, but at least one of the messages that we were able to distinguish or to highlight was that we found ethylene biosynthesis and signaling pathways activated very shortly after inoculation. And this was partially paradoxically, but because as you know, ethylene is an inhibitor. It has been a classical and known from years that is a classical inhibitor of nodulation and symbiotic interaction. So how can we explain this apparent contradiction? Indeed, it was identified in some of the pioneers, researchers in Spain in the field of nitrogen fixation, that actually, based on ethylene evolution measurements, it was observed that as around 24 hours after inoculation, there was a burst of ethylene production. And the thing is that it was related to a general defensive response. And this is here where we can magnify maybe this idea. Because having a look at the data set that we generated, and if you look at the specific markers for ethylene biosynthesis, for ethylene signaling in these samples, what we can see is that there is an ethylene response that occurs really early. And we are talking here about 1 to 3 hours after inoculation. So that is even earlier than non-factor dependent responses. So what we are observing is maybe a first burst of ethylene signaling occurring just at the contact of the bacteria. So this is really not factor dependent, and is totally AIM2 dependent, because you can see AIM2 mutant behaves, the expression pattern behaves totally different. So we see this ethylene biosynthesis activated during these early times, and this occurs in a non-factor receptor dependent manner. We also have a double mutant here that is DOMI1 sickle. And now we have RNA-seq data also from this double mutant that again is let's say a comparable, or like another proof, independent proof that indeed these responses depends on the active not-factor signaling cascade. Okay, so what about ethylene production, you may say, because we are talking here all the time about expression data. And we do actually, we were not the only one wondering about this. And actually in Lotus japonicus, the method to detect using laser-based techniques, the method to detect ethylene evolution in seedlings is being set up. So there was this work by Dougal Wright in Jens Stuckart lab who actually tested this, and indeed they saw a correlation with inoculation and production of ethylene in these plants. And this is totally dependent on compatible rhizobium, and even using isolated NOS factors, they could see this ethylene evolution. So now the question is, okay, this is clear, but actually what do we know about the ethylene biosynthesis and signaling pathways in medicago? And the response is that we know little, and most of what we know is basically based on what has been described for Arabidopsis. So regarding the biosynthesis pathway, you know, it's very simple. There are two enzymes that are working for the biosynthesis of ethylene. The first is ACC synthase that synthesizes ACC, that is the precursor of ethylene, and then it acts in combination with ACC oxidases that generate the final product that is ethylene. And it may have the same as it would have happened probably to you as well. If you are interested in knowing which are the gene members of these families, what we all do, right, is to go to the genome and try to check that based on functional annotation. And when I started this work, that was quite a couple of years ago, well, a couple of, more than a couple. The genome versions were not maybe optimal, the annotation in these genomes versions. Now with the new release, with the version 5, the annotation has been improved, but still there were a number of genes that were wrongly annotated as ACC synthases and mostly ACC oxidases. So just to draw a note of caution, when you see some functional annotation, just double check that those are actually ACC synthase or oxidase genes. And to clarify a bit this, for a while I had in my mind the idea to get this work together and try to analyze using phylogenetics and a bit more of a in-depth work to try to identify which are real, at least based on structure and localization of conserved domains, which can be more, say, reliable, called ACC synthases and ACC oxidases. We did so for Medicago truncatula, for both A17 and R108, and also for Lotus japonicus, again, the two main cultivars, and also for a couple of other species, legume species. This has been summarized now in this review that has been accepted recently in Journal of Experimental Botany. And what I can... So I hope that this will be a reference for the community. So if you have a doubt, you can go there and check whether the gene that you are interested in is close ortholog to one of those. And we also collected and compiled data from different expression data sets. And one of the ideas that emerges here is that there are several ACC synthesis isoforms acting in different tissues and at different levels during this early interaction, and one ACC oxidase at least that we see a differential expression clearly. So it's not that easy, but we are working there. What about ethylene signaling? Well, ethylene signaling is a bit more complicated than the biosynthesis pathway. Just to refresh your memory, the ethylene receptors are located in the endoplasmatic reticulum membrane. There are several of them that forms dimers. So in the absence of ethylene, there is a negative regulator of the signaling pathway that is basically based on this protein, CTR1. And what it does, it phosphorylates AIM2, that is the protein responsible for the sickle phenotype that we obtained. That is phosphorylated at the C-terminus domain. And when it phosphorylated, this is sent directly to degradation in the proteasome. The same happens to other regulatory proteins. I didn't recall them in the previous slide, but those are AIM3 binding FVOX proteins that are regulating these downstream responses to ethylene. So in the absence of ethylene, all these regulatory proteins are sent for proteasome degradation. That's the idea to keep in mind. When ethylene is present, however, there is a conformational change that occurs in these receptors, and CTR1 then stops its activity, its negative regulation. So this leads to the cleavage of the C-terminus domain of AIM2. So it's translocated to the nucleus, where it interacts with other proteins to activate ethylene-dependent transcriptional responses. But very interestingly, in recent years, it has been shown that this C-terminus domain not only travels to the nucleus, but also stays in the cytosol. And what it does, it's also able to affect the translation of several regulatory proteins, such as these FVOX proteins that I mentioned. Again, this is all known in Arabidopsis, obviously. But we have this dual option of regulation to keep in mind. So summarizing what we know or what we are still trying to understand in these early symbiotic events. So upon bacterial contact, there is this quick burst of ethylene occurring very early. Those are the two FVOXes, or at least two, there are more probably. But two of these similar to the ones I mentioned in the slide before. So we have this early ethylene signaling activated as soon as 1 to 3 hours upon inoculation. Once we have a rhizobium infection, there is an activation then of the biosynthesis pathway. But now the question is, how does this occur at the molecular level? And this is what we are trying now to elucidate in collaboration with groups in Wageningen University, with Wouter Collen, and with Fernanda Carballo-Nivel and Andreas Nivel in RAE Toulouse. We are trying to detect or to have candidates to this direct regulation in the promoters of this ACC synthesis that are activated. So this is work in progress. And also then we will need to close the loop, let's say, and fill the gap of what about these ethylene AIM2-dependent signaling mechanisms that still we don't know. And to do so, I am also collaborating with Maria Eugenia Zanetti in Conitec in the University of La Plata, who is a specialist in this polysome dynamics and regulation of RNAs. So we are trying to investigate which are the other effects of this, the C-terminus domain of AIM2 in the cytosol. Sorry. So that is most the molecular, let's say, side of what we are doing. But I had also one question in mind that is that was also a bit controversial in the literature, and is whether we do. So the question is, do we need AIM2, an active AIM2, to generate a nodule and to maintain and sustain nitrogen fixation in nodules? And to do so, what we did was characterize the response of sickle, our favorite mutant so far. And another question that I had is, well, ethylene is a well-known hormone, a stress hormone, right? Stress related, we all have this in mind. And it has been often associated to drought responses. So one question that I had was, can we see a differential effect of the drought response that we have in sickle compared to the wild type? And this is work that we have been doing recently. I don't have time for the details, but we are working on a manuscript preparation right now. And as a spoiler, the thing is ethylene, so sickle is able to fix nitrogen. It develops nodules. Obviously, they are much smaller, but they are fully infected. Nitrogenase is active in these nodules, and we don't see any effect of defect in terms of nitrogen deficiency in those plants. So they can rely 100% on nitrogen fixation. And then the other question is that, to our surprise, the response of sickle to drought stress was not as different to the one we thought, the one we typically observe in A17. But we're still trying to identify why, and we have a data set to try to get to a conclusion. Okay, so just very briefly, the two lines that are a bit different, let's say, in our laboratory are the ones that are being carried out by our postdocs. Lucas Castellani, in collaboration with Marie Merous at the University of Perpignan. What we are trying to explore is quite a novel field in plants, and that is the putative role of the potential role of extrachromosomal circular DNA in drought responses. So we are trying to first check whether we are able to detect these extrachromosomal DNA molecules in Medicago truncatula. We are testing different tissues to do so. And we are then asking whether drought stress may play a role in the expression of these regulatory molecules. So this is, again, something we are very excited about, and still is work in progress. And finally, Isa, she's an expert in plant growth promoting bacteria, and her research project is focused on trying to isolate local community bacteria living in the rhizosphere or in close contact to Medicago sativa, Medicago truncatula plants, trying to identify PGPR traits in these strains and trying to produce synthetic microbial communities in order to test their ability to help plants to promote growth and to have a better drought resilience. So with this, I would like to thank you for your attention, and I am always open to support either PhD applications or postdoc applications. As you have seen, we have quite some experience in the Marie Curie Fellowship, so feel free to send me an email if you have an interest, and I'll be happy to reply.
Rafa, I think you are still muted.
Yes, true.
Thank you, Naomi. So thank you, Estibaliz, for your very nice talk. So now is the time for questions. So just a reminder, if you have entered to the platform as with your microphone, you can switch on the microphone and make the question. If you have entered as listener, you cannot do that. So you have two options. You can write down your question in the chat or go go out and go in again with your microphone. Okay, and I accepted that. So I don't know if anyone has a question for Estibaliz. So you can also switch on your camera if you want to make the question. Well, in the meantime, if someone wants to have a question, I have one. Well, I have a couple of questions, but you said at one moment, no, ethylene is an inhibitor hormone for many processes, including ovulation. But how is that in a dosage dependent? So because we are working with volatile compounds, one of the volatile compounds that some microorganisms release is ethylene. And we don't see an inhibitory effect of ethylene in this. It's a mix, no, of ethylene and other volatile compounds, but we don't see. Actually, what we see is a growth promotion with this mix.
You mean using GPRs or what?
I mean, in PGPR or PGP, if we work with fungi, we see that the ethylene doesn't have, in the concentration that they are able to release the ethylene, that normally is, we can detect it, but it's lower than the one we use to apply with ethefone or other molecules, other systems. We don't see this inhibitory effect. And I don't know if you have any idea about nodulation, if there is, maybe there is a positive effect with a lower doses, before do you understand the question?
Yes. So, I mean, I think there are two very different words. So one is the endogenous levels of ethylene that are definitely very low, and also I think we need to keep in mind that being a gas, it acts at the local level. So and then as you can see, there is a discrimination in different tissues. So yeah, that is something to take into account. There is obviously hormones are very complicated to work with, so and there is interaction with other hormones as well. So the doses are definitely important to your experiment. And so the application of external sources of ethylene and what is going on inside are two different words.
Yeah.
So in my opinion, so yeah, in this interaction, in the legume symbiosis, rhizobium symbiosis, it's very clear. It is inhibitory. It is true that it also acts at certain time points. So there is a gap in which these inhibitory effects are kind of ignored, and then again it starts a new cycle. So yeah, it's a complicated.
No, I guess so. So related with that, do you know if the rhizobium bacteria are able to produce ethylene? I have no idea.
No, I think in general, I mean, it has been shown in some PGPRs that there's a trait that is actually the ACC, the aminase, I think. So the idea is that some PGPRs may help colon or yeah, I'm not an expert on this, but I think it facilitates their colonization if they produce this ethylene degrading enzyme. But I think in rhizobium, this has not been.
Okay.
Yeah, I'm not particularly confident that this.
Yeah, I have no idea. So it's just a curiosity.
Yes.
So you have more questions for Estibaliz?
There's something in the chat.
On the chat.
No, it was a question.
Yeah.
Well,
Yeah.
yes. So no questions.
Well, maybe it was so clear.
I hope you followed all right. I was a bit quick because of the time limitations, but I hope it was clear enough.
Yeah, I think so.
Okay, good.
Yeah. So I have more, but maybe we can discuss in another moment. Okay. So, I don't know if there is no question, we can switch to the second speaker.
Yes.